Hello?
I heard you.
There we go. Yeah, my microphone didn't work before.
It's weird because it almost sounded like I could hear you like attacking on your computer, but not your voice.
Oh. Well, yeah, sometimes the microphone doesn't quite pick up, so maybe it's picking up something else. But anyways, okay, how are you?
Oh, right. I'm sort of just stopping by in the midst of things. I'm actually mostly attending the IEEE Coins conference today.
Okay.
It's a lot of it is quite hardware technical and not, a lot of it's very engineering oriented,
Right.
but I'm kind of scoping it out for some other specific reasons.
Yeah, that's great. That's great. So, welcome to the meeting. This is the end of the GSOC period, so I know that we talked last week, Mainak gave a presentation on his work, and so that's been submitted, and I now I owe the program an evaluation, which was excellent. So I'm looking forward to doing that. I'm going to talk about a number of things today. I don't know if other people are going to show up, but we're going to go over the project a little bit. We're going to go over some other things that related to the project, maybe next steps in terms of what we want to do with the DevoLearn project. This is the broader project of this DevoLearn platform. And then we'll get into some papers. I don't think there are many other submissions. So hello, Mainak, how are you?
Yeah, I'm doing good.
Good. Glad you could make it. I had just about to start talking about DevoLearn and the project. So did you have any problems submitting your project?
No, the submission went smoothly. I'm actually done with the submission, so it went well.
Okay, that's great. Yeah. And so I will get your evaluation done soon. And then we'll move on to next steps on the project. So that, that involves like the paper and things like that. So let me share my screen. All right. So this is the project 3.1. This is the thing that Mainak issued a pull request a couple days ago on this in the DevoWorm GSOC 2021 repository, and this is basically the official work repository link, the PyP package. So this is on Python P, which is PI, which is the it's like a place where you can download packagers for different Python-based open source software. There's the web app, and then the web code repository. And then this is the README, which has a lot of the stuff here. It's broken down into the different phases of the project. So this is segmenting nuclei, defining the data set and data loader. This is the model here. It gives some nice detail here, model performance, upgrading the cell membrane segmentation model, fixing the flawed data, the training metrics, deploying DevoLearn models on the web. So this is the web architecture for that. And so yeah, we're right now. I guess it's on. This is hosted on Heroku app, correct?
Yes. Yes.
So then, okay, and then, but of course you need to handle all of the model and the data. So this is where this comes in here, and then. So then you go through we can step through the web app, and then interactive plots, and then the weekly blog posts, which if people are applying for GSOC next year, this will be useful to see how to do this. So let's go up to the official work repository. This is our repository we're in. This is the PyP package here, which is actually a DevoLearn/DevoLearn. So this is the PyP package as it exists on their site. This is where you can download the package, install it on your own computer, and run it. So this has every feature that you would find in the web app, but you can download it onto your computer and run it. And there's some good news on this front. And I know Mainak tweeted on this a little bit and put this on the Slack. But we have DevoLearn has just hit 10,000 downloads via PyP. So this is the interface I was just showing, with over 800 downloads in the last 30 days. So that's really nice. That's a good user base potentially that we're building on this. So this is you can look at the stats for these PyP projects, and I know we looked at it in an earlier, I think in February, where Mayok had brought up the stats for it, and it was starting to grow then, but it looks like it's grown even more now. So we've got where we're getting a critical mass. So we're at 10,000 downloads, and this was a couple days ago. So congratulations to everyone involved in that. So that's the main page there. This is the web app. So I know Mainak is showing this quite a bit, but it's been kind of work in progress. I think last week he showed us a version of the final product. It'll take a while to load.
Yes, it does take a while to load because it's actually free, so I guess that's the downside of it.
Yeah.
But yeah, it actually works. Once you load it up, it works.
Oh yeah. We had we also have the OpenDevoCell, which kind of does the same thing. It's also hosted on Heroku app, and it's a different model, but it's very similar to this. So this is DevoLearn at herokuapp.com. And so we have choose one of the following here, which is home, and you can choose your type of segmentation or lineage prediction that they want in your analysis. So this just kind of goes over the detail of it. And then we also have some links to data sets here. The EPIC data set, the cell tracking challenge. One of the things we didn't do in the GSOC period is talk about the DevoZoo platform and or the DevoZoo repositories and how we might incorporate that. But I think that's maybe down the road a bit. The DevoZoo, of course, we know, and we've talked about this in, maybe several months ago now. It's been the last time we've talked about it. It's these different model organisms for development. And we basically have got sort of curated a number of data sets for each organism. So it's not just C. elegans, it's Drosophila, it's spiders even. We have some other types of embryo zebrafish that have like this, we have some data that people can download, either microscopy data or data, something like the EPIC data set where you have images and or cell tracking data. So all those things are available. Now, we didn't really integrate any of that we've been doing some of that integration with the models. In 2020, the GSOC project, we did some work on that then, but so these are data sets that are probably the most relevant to this model though. And so you can try those out. But also, if you're a neophyte, or a beginner, you might want to try one of these. So for nucleus segmentation, and I think you have some base data here that you can use. So you can actually just put your own imaging data, or you can take your image data, image by image, and put it in a file on your laptop or whatever, and you can upload these images, and then you can pull them up and it'll segment them right in this interface. And so you have to limit your file to 200 megabytes per file. A lot of most microscopy images are not nearly that big, so you'll be able to do quite a few images without overloading the system. But so this runs on this image, which looks pretty noisy here, but you get a nice image here. And there's also a threshold slider on this side, so you can increase or decrease the threshold. So this is just like kind of like filtering out a lot of the background noise, or treating things in the background, or treating things that are not like extremely obviously bright and sharp as a cell body as background noise. So you have this, if you go to like no threshold, you'll get a lot of noise in the image, and you can see it's carrying over from the noise here. But if you go up, you increase the threshold, you get this, which is a lot cleaner. But there's a trade-off here. If you really set the threshold high on some images, you will need to set the threshold high because there's going to be a lot of masking of noise, but you're going to lose some details. So this is something to keep in mind if you're using this. And so you can do this for any of these examples. This is the lineage predictor. So this is from, this example image. You can also upload your own. And this is a C. elegans embryo. And this pulls out just for, I guess this is just set up for C. elegans now, because different embryos of different lineages, different classification schemes. But basically you have these different classes, and it's able to pick out, based on their location, kind of what cells you have in here of what lineage. So this is nice. It kind of works with the labeling that we already have for C. elegans, and of course that works in C. elegans. In other organisms like zebrafish, it might not work so well because we have a different scheme of lineage, cell lineages, plus there's a lot of what they call regulative differentiation, which is where cells just kind of divide, and then they get take on a fate based on the chemical signaling around them. So C. elegans, that's typically not the case. You can tell what a cell is as soon as it's born. So this is a nice feature. We might, in future years, we might work on this a bit. We might work on linking like a classification scheme to an image array like this, where you have a bunch of cells in an image, and trying to figure out where the what the lineage is without necessarily a label. So what I mean is, if you have this embryo and you can maybe take a bunch of images in a time series and watch them divide and start to, this cell might give birth to like 16 cells around it, be able to track that lineage out and just build like a lineage tree from just the images instead of like the labels. Now that's not particularly useful in C. elegans, but it would be useful in like, in something like mouse, or something like a induced pluripotent cell, where we have cells that just divide, and there's certain lineage properties of those cells in that region, and so be able to like identify like a founder, what they call a founder cell, and then find its descendants and track that. That would be nice, but that's beyond the scope of this year and maybe even next year, because I don't necessarily know how we would do that. So that's basically maybe what we'd have in the future. So I don't have a diagram for. Oh, hello, Akshay.
Hello.
Okay. Oh, okay. Mainak has something to show us, so I'll wrap up the DevoLearn stuff, and then we can get into the droplet stuff.
Today even I have made some progress.
Okay. Yeah.
I'll show after Mainak.
Okay. All right. Good. So I don't have a map of this, but basically we have the Open Devo cell model, which is not a pre-trained model, but it's a machine learning model for, I guess it's general embryos, but was trained on C. elegans, and we know it works on C. elegans. We can use it for different things. We also have the DevoLearn platform, which has in 2020 we started with the basic pre-trained model, and this year we've augmented it now to have this web interface, and now we can actually just do this. Now we have the Open Devo cell and the DevoLearn platforms that are both two different types of models, and they're both web interfaces So now we can actually just go in and analyze data in two different ways. And so now if you don't know what you're doing, or you come to the group and you're like, I want to, try to analyze some data to get started, now we have some really nice tools for you to use. And people are downloading this, in the general public. So hopefully we get some feedback from that. Now, speaking of which, I wanted to go over maybe next steps. So the next step, of course, and this is something that we were working on last year, we put it aside, is this DevoLearn paper. And this is a preprint paper, which we were trying to submit it to the Journal of Open Source Science, but they didn't really think at the time it was suitable for that venue because it wasn't hadn't been used enough, and so we didn't do that. But we have the preprint, and I'm not going to show it because it's not really ready to talk about really, but it's a basic outline of the DevoLearn platform, which is this software. And of course now we'll have a lot more to add, and it goes through, kind of walks through what it is, what it does, talks about a little bit about pre-trained models, why it's relevant to what we're doing. And then, of course, if you're not familiar with the DevoLearn platform itself, it's not just the software. Let's see if I can get to it here. No. It's actually this whole platform on GitHub. And it's this educational platform where you can download it, and I think this is where it is, right? Okay. So this is the DevoLearn platform here. Let me show you. Okay, so this is the DevoLearn platform, and so you have the DevoLearn/DevoLearn repository, and this is where the software is, and this is where you can download a lot of the code and everything. We also have an education location. We have theory building. We have the C. elegans DevoLearn, which is a broader web app that supports a lot of the web aspects of not only the DevoLearn software, but the larger DevoLearn platform. We have data science demos, which we've had a number of people contribute. So we've had Mayoka's contributed, Krishna's contributed, Ujwal, Shruti, Vrudik, Mainak, of course, and Malvi. So they've all contributed something to these data science demos, and there are different things in here that if you want to look at, like GANs, networks, centroid extraction, this one here, which is a command line basics in Python. So there are a lot of different tutorials in there that you might want to look at if you're interested. I know that Jesse and I were talking on Saturday at our other meeting about demos for different things. And this actually, I forgot about this, but this is focused more towards data science for biology, but this is another way that we might do these technical tutorials. This is something that's fairly simple for a student to do. That's a network data set. This needs to be updated in terms of organizing things, but I think this is the DC GAN digit. So this is a nice, this is something Mainak put in as a tutorial, and it just kind of goes through, I think this is for GANs, implementing a GAN. There's a Colab notebook and everything, so we can, you can go through that. So this is the platform, and I've been presenting on this to a number of different groups, emphasizing the DevoLearn software, but also the larger platform. And so this is, DevoZoo organisms. So this is the DevoZoo. There's actually a website skin to this, but these are the different repositories for the different organisms. So, okay, so this is Akshay actually, who put in something in axolotls.
Yeah, that's me.
Yeah. And, there are other organisms like ants and spiders, which we don't really have any content for right now on this. Basilaria, which are the diatoms we talk about. Siona intestinalis, which is a marine invertebrate, the simulated embryo, which is where we numerically simulate an embryo in terms of its cell tracking and things like that, and then zebrafish, and of course C. elegans. So this is the Devo Zoo where we have all these different organisms. So this is sort of the future for DevoLearn, and we want to kind of develop this out a bit. We definitely want to write up a paper on this on the software, and then keep talking about our keep talking about this to groups so that they thank you, Jesse. Talking to groups about this so that they have some idea it exists and that they can use it. So now, Mainak, you wanted to present on your topic?
Yeah, I'll just go ahead and share my screen.
All right.
So this work is actually regarding a mail that I received from Dr. Gordon back in the day when I was starting GSOC. So I couldn't really start work on it back then. I started work on it today. So I'll just go ahead and share what the progress is. So can you see the screen?
Yeah, it's very blurry, but yeah.
So is it okay now?
Yeah, I can see it.
Yeah. So see, the code it's not really important, but I'll just go over what I have. So this is the input data that I got from him. So I'll just go ahead and zoom into the image. So this shows basically 4 photos which are taken in 4 different time points. So this shows these droplets which are changing shape with time. So at this point they're really small, and then they form hexagon-like shapes, and then they become larger, and then they form into triangles and lines. So what the goal is to basically find out the number of shapes that we're getting. So what I did is that I cropped into this third image right here, which is this, and then I'll just show you the results that I got. So this was the input image that I had, and then I could basically map out the shapes in this format, like the borders of the droplets. I could use some OpenCV based transformations to get this map, and if I draw it up, and if I draw that on top of the input image, we get this. So it might be very obvious that a lot of these shapes are being missed out because there's a threshold that I've placed on the minimum area under which the shapes would not be detected. So if I set the threshold to be a lower value, more shapes would be detected, but that will also pick up a lot of the noisy parts. So like this part right here, this might not be it might be something else. It might just be noise. Or these lines right here, they could also be seen as shapes if the threshold is set to be very low, the threshold of the area. But if I set it to be high, it gives out this result. So this is actually still a work in progress, and I'll be working on this in the upcoming weeks. So yeah, that was it for this update.
Oh, that's nice. So the numbers represent the different shape that you found. Like you found a unique shape.
Yeah. So 5, it basically shows that the shape of this droplet has 5 corners to it. But as you can see, it's not really obvious that there are 5 corners. But somehow, like, it roughly comes out to 5 edges actually. Like it's a bit rounded, but it's actually a bit blurred out. I wish I could show you a more clear image. So maybe I'll come up with that in the upcoming weeks.
Yeah.
And yeah, the numbers basically show the shape. Like 6 means a hexagon, and 5 means a pentagon, and 4 means a square or a rectangular shape. So trying to detect the approximate corners.
Yeah. So this is what algorithm are you using for this?
I'm using a lot of OpenCV based algorithm. There's this thing called I'm using the contours, and I'm also using some Gaussian blurring, and then thresholding is also there to binarize the image, and then there's also a bit of K-means detection at work. So it's actually a mixture of a lot of OpenCV based algorithms. So I'm trying to find out the best match to get the shapes properly and to get to detect everything properly. So yeah, that's something I will be experimenting on in the coming week.
Well, nice. Thank you. Yeah, that looks pretty good. I mean, being able to classify the shapes, and well, maybe we can catch up on update us on this in future meetings and see how that's going, and
Yeah, sure.
Nice.
So now maybe Akshay can go ahead and present.
Yeah, go ahead.
Yeah, am I audible?
Yes, you're audible.
Yeah.
Okay, so I'll try sharing my screen. Is my screen visible right now? Or?
It's still gray. I'm gonna reshare it.
No, for me it's showing I'm sharing, but I think this is the problem with Firefox.
Oh.
Maybe you can try Chrome also.
Yeah, let me just come back right now.
Okay.
Yeah, this sometimes actually happens in this platform.
Yeah.
Jitsi platform.
So, Mainak, what are your plans for the fall? Do you have anything you're doing? Are you attending school this term?
Yeah, I will be attending school, so I'll be sort of busy with that.
Oh yeah, yeah.
Is my screen visible now?
Still not. I still see it gray. Are you? What are you sharing?
Actually, is it a collab notebook, or maybe you can send me the GitHub repository link then?
Well, it's actually a messed up notebook. I'm not maintaining it well. It's just I'll try to share the Plotly link if that's possible.
Yeah, that would be good.
Or maybe you could also send us the collab notebook. Then maybe we could share
I
our
will do
screens.
that. Yeah, let's do that. Wait. Yeah. Let me share the link.
All right. Okay. I'll share my screen on this.
Yeah.
All right, let me share my screen here. It opened up. So this is for Akshay. So here you go, Akshay. Let's walk through it.
I would like you to scroll down. These are the first parts, pre-GSOC. I mean, like I was trying to segment and canalize detect all this stuff. But scroll down, just scroll down. The last quote.
This part?
No, the next one. The next course. Yeah, this one.
Okay.
So,
Oops.
this.
There we go.
So, the thing is I actually gave some math in my, my proposal before. So the thing was I wasn't sure. I had to research a lot to find some sphere polar coordinates and mapping it. And the thing is, right now I managed to make a 3D sphere, which is actually great. Now I have to find a way to display the Axolotl embryo dataset. That is in the paper it's given A, B, C, it's given in an order. So I'll have to figure out which goes on the bottom, and it's given in the paper. So right now I was able to make a mesh grid and a sphere, but I am actually having trouble bringing the dataset images onto the surface of the sphere. And there is this parameter called surface color, which is supposed to do that, but it's not doing it. So I was actually figuring it out till now. So I guess that's it. Small progress, but so the thing is, you only see this blue thing. I don't know why it's happening. It should actually show the dataset image, which I'm loading. Just one image I'm trying to spherize right now. I'll have to find the coordinates of the images and map it according with the mesh grid, which I just made. You can see theta comma phi equal to np dot mesh grid. So I'll have to try and do that, but I don't know. It is in my mind, but I don't know how to do it on the system. So I mean, I'll try doing that by next week. So.
Well, good. I mean, this looks good. I mean, this would be a good model for it, so it's going to generate coordinates. So wherever it is on the image, you'd be able to look at the coordinate Yeah.
Basically, I was actually not sure of my math like when I submitted the proposal, but I mean like I had to go through so many papers and I had to try deriving one like equation on my own, so I could actually verify that it will happen. But yeah, now I actually tested it out by coding it, and it's actually working. So on to the next step, that is like but one doubt I had, Bradley, is like wait, I'll share you my proposal doc also so that you can view it. uh, I actually had some image doubt I wanted to, yeah, this is okay, yeah.
All right.
So if you yeah, scroll down, um, yeah, you see this like convert the data set.
Okay.
This image,
yeah, here.
Yeah, so A, B, C, D is like if you just scroll up one more time, I think
Like here?
it's written. Yeah, it's written like yeah, A flip to the bottom, right? So the this is what I had in like as a doubt. Like 8 images would supposedly form a sphere, right? Like according to the research paper, which is
A sphere or like a 180-degree view. I guess. Yeah, I guess.
I
It
think
would be,
100.
Yeah.
Is it 180? That was my doubt, basically.
Okay.
Basically.
So yeah, if you have the sphere, and you're looking, say like you have the globe of the Earth, and you're looking at the North Pole, and this image A, you're kind of looking down at the Earth from the North Pole. So you have like different continents, towards the pole. They're right kind of in the middle.
So A is the South Pole. What you're saying?
A would be the North Pole for this, and then you'd have the North Pole and all the land that's near the North Pole. You could see pretty clearly, and then at the edges it starts to deform as you move towards the sort of the edges of like maybe towards the equator. And then what's happening here is that the Earth is flipping, so that you're flipping along an axis. So you're maybe flipping along, going from the North Pole, and you're going across maybe like Europe and Asia, in that trajectory. So now the Earth is moving, and you're started. The top view is now like Siberia instead of the North Pole, and then later it's maybe India, and then further, and then it's starting to move south. So you're
It's actually
seeing
moving downwards, yeah?
Yeah.
Like from north.
And so
Okay,
then
so
H would be
so-
the South Pole. So you'd be
okay,
getting
so
to the South
E would also be the South Pole, if I'm not wrong.
Pole.
Yeah, we have Susan now, and she's
Because I don't know, E through H, or is that like the rest of the image, or like because it's flipping? I know it like goes from one orientation to another.
I think Susan can answer this because she's the author.
Yeah.
Of course. Yeah.
Yeah.
Hello, Susan.
Oh, she's here.
Yeah, she's here. She
All
just
right.
joined. That's
All
right.
right. Hello, Susan.
Hi.
Yeah. Welcome back.
What was the question? Yeah, hi.
Yeah, Susan. This screenshot I actually took it from your paper. So you can see, right, like, so what I've actually copied the statement which was also written over the image in the paper to my proposal. So you can see, right, like A is the flip to the bottom image. So I actually had a doubt whether it is the, so if you are considering Earth, it would be the North Pole or it would be the South Pole.
Or the top
If
of
you have the North Pole up, then the South Pole is down.
So it would be the South Pole.
So it would be like the South Pole.
So B would be moving towards the equator, like from south towards north, yeah?
Yes.
Okay.
And C would be the equator and D would be Europe. I mean,
Or something. Yeah.
well,
The question.
and E to H would be the North Pole, yeah? I mean, nothing.
Yeah, that's all North Pole.
So,
Yeah.
so there's no entire sphere. You just have this 180-degree view.
It flipped. My camera doesn't take pictures fast enough for some of the eggs to have a change in E to H.
Okay.
But yeah, I need a mirrorless camera to make this work better. And that's why I did that 3D sphere. I figured it would work better if you could just take, if you could just leave the eggs sitting still. So that's why I made the other system.
Okay, so yeah, basically I had this doubt because I was trying to visualize this in mind and it was just getting out of hand, you know.
Yeah, so you probably just use the first 5 images to work with.
Yes, that clears a lot. Bradley, could you show it?
So
the
down
sphere.
this one here, the converted.
No,
images?
the sphere.
Oh, okay. Yeah, this one. Okay.
Yeah. So so this is also just a basic implementation, but right now I would have to bring the dataset images like from bottom and like you said, from moving from down to up. So it would. I'm actually confused with the Plotly libraries, but so far now I have got a clear idea on what to do. Thank you both, Bradley and Susan.
Okay, and I'll get some pictures with that new
Yeah.
The
Exactly.
cameras will be always in the same spot.
Okay.
The egg won't
Okay.
move.
Okay, Susan. You let me know once that's done. And
I need to automate it. I need to get my microcontroller out and get it to work really.
Who made that? Like the Arduino and just a question. I mean, like, when I read it long back, the paper, I don't remember it clearly, but I remember Arduino being used. Yeah.
Yes, it was an Arduino, and I had a student on the project, and he did the programming for it.
Okay. Wow. Yeah. Okay.
I've programmed Arduinos, but not very much, and he was actually good at programming, very good at programming. He actually went on to get a computer science degree. One of the success stories.
Wow. Okay. Yeah. Well, yeah, that actually clears it. So thank you. Yeah, correct time you joined. I mean, I wasn't expecting you, but yeah, you just joined it.
I had
Save one more week. Yeah.
I had a guest here, and the guest left, so I was able to come on board.
Well, thank you.
Yeah, thank you. Hello, Shruti. How are you? And of course, Susan. Well, I think you're muted. You might be having a little trouble with the mute button today. Yeah, it's fine. All right, yeah. Well, thanks for joining, Susan.
I'll mute and everything.
Oh, that's okay.
Like that.
All right. So, yeah, that's what Akshay was showing, this work that he's been doing on the Axolotl. He was going to tile this with image, but it's not visible right now. But the idea would be to tile this reference frame with images, and then be able to flip it. So you can do this to explore the space, and the features on the image would have some coordinate system attached to it. So, like right here, I can get a coordinate measurement. This part of the sphere versus this part of the sphere. So that looks good. I look forward to seeing the output of this, and this is, of course, some of the image processing that was going on with the image.
That's embarrassing code.
Okay,
That's why I hesitated to share the collab link, but yeah.
It's okay. Yeah, it's just a work. It's always a work in progress. So,
I'll make sure that I get the images with the new microscope as soon as I can.
Yes, Susan, take your time. But I just needed to implement this. This was the tough part. Now I just need to give the images on the surface. Yeah.
yeah.
All right. Well, thank you for doing that. I hope we can get some really nice images of eggs.
Yeah, look forward to it. So let me go back to the submissions document. So we were talking about the DevoLearn paper, and that's something that'll be probably on the front burner now that we're done with the summer period. So this will be like updating the paper and then pushing it out into a preprint server, putting more stuff in the paper. So be on the lookout for that. I've been working on this non-neuronal cognition paper, so that's coming along, and I didn't bring the paper with me to the meeting today, but it's getting close to being where we want to maybe submit it to the special issue. I want to go over it in the meeting and get some feedback on some of where we are with it, but it's not quite ready yet for that. So all that's going to happen probably in the next few weeks. And then we still have some of the outstanding issues here. We have this boring billion idea for a set of book contributions. We have the Kindle book, and I talked to Krishna about that, and he says he's still working on it. So that's still sort of in the works. This mathematics DevoWorm, I think I showed that last week. That is something that I've been adding to. So we not only have these different network models and spatial models and von Neumann neighborhood models, like this is for a cellular automata, but we also have these other models. This is from a paper we did on game theory applied to embryos. This is this differentiation tree work that I showed a couple weeks ago. And then this category theory model, which is actually from another paper that we put out earlier this year. So I'm going to try to work this into a short paper. So we have like these different models, and then we have them in front of us with sort of their importance. And then, we'll go from there and see where we can put this. Maybe, make it as another preprint, or maybe put it in some sort of journal. I think it's a nice resource because there's nothing like it. I think this originally started when I think we were in the OpenWorm group, in the group of senior contributors, we were contemplating like that worm book, which is this reference for C. elegans researchers, why they didn't really have any computational information in it. Like it was all biology and there was no computational stuff. And I don't know if this would be a good fit for worm book, but it definitely, I think, would be useful as a reference for people. Obviously, maybe not in this form, but in a more research-friendly format. So we have these other things, the test of Williamson symbiosis, the molecular level simulations, the diatoms. Those are kind of in progress. And of course, Mainak showed us his work with the, where is this thing? Oh, the shaped droplets work. So I know Shruti and Susan weren't in the meeting at the time, but he's been doing some work with shaped droplets and categorizing some of the droplet shapes and, trying to analyze it by the number of sides. So it looks pretty interesting. We'll see where that goes, and maybe we can move that forward as well. And then finally, this book by Steve McGrew, The Eye of Nature, that's still in the pipeline. So I don't know what we'll do with that, but we're kind of still thinking about that. So that's all I wanted to talk about with respect to that. Now I want to move on to papers as usual. So we have a lot of things in here today. I'm trying to think of where to, I guess if Susan's here, we'll talk about liquid crystal biology. And I wanted to kind of go over some of this. First of all, I sent Susan this book and I put it in the Slack channel for DevoWorm. And this is a book called The Physics of Liquid Crystals. And this is Pierre Desgenais, who's a French I think, chemist, physicist. And this is from 1974. He wrote a book called The Physics of Liquid Crystals. And so I think if you've heard of liquid crystal televisions, you think, well, what does it have to do with biology? And the answer is well, it's biophysics. Basically, there's this theory of liquid crystals. Liquid crystals are these molecular systems that behave collectively, and the idea is that as they behave collectively, they exhibit a lot of these properties that you can classify and that describe sort of these broad behaviors. And people have done this. I was at a workshop on this a number of years ago where they were doing this in bacteria, looking at bacterial rods and how they orient themselves in a culture dish. So there's a lot of applicability of liquid crystals to biology. But back here in this instance, this was largely an exercise in physics and chemistry. So, this goes through a lot of things. The study of liquid crystals is complicated because it involves several different scientific disciplines: chemistry, optics, mechanics, more specialized tools such as nuclear magnetic resonance imaging, and a certain sense of vision in three-dimensional space in order to visualize complex molecular arrangements. So this is the sphere that Akshay showed is just like the beginnings of this. You can have much more complicated topologies to put these phenomena on. So, if we want to explore, say, for example, the edge of a sphere, that's one thing, but then there's so much structure, within the sphere itself. And here I'm talking about an embryo, but this could be any kind of system. And those are the kind of things that really, if you really want to get a lot of detail, you have to be able to do things like fly through the environment or look at it from different angles. So that's why that kind of approach, where you have things on a sphere, where you have things where you can rotate it, is useful, because it's, depending on how you look at it, or, depending on your perspective, you can analyze things a lot more effectively. And of course, this was right at sort of the beginning of supercomputing. So as supercomputing has grown in power, we can do a lot of that. We can simulate things virtually and, see things we've not seen before. So, in liquid crystal biology, we have these building blocks here. We have small organic molecules, long helical rods, and associated structures. We have these different, I think I talked about this last week or the week before. We have these three different phases where you have pneumatics, cholesterics, and smectics. Ranging from ordered to disordered and different modes of order. So you might have a bunch of bacterial rods on a single plane, and they're either all oriented in the same direction, or they're oriented in different ways that give the plane some order, but it doesn't necessarily look like order at first glance. So they have something called long range order. And so these are all sort of physics terms. People think about this a lot in the literature on self-organization. They think about how things are organized at these different length scales. So whether things are organized locally or globally makes a difference in how the collective behaves. And so this could be cells, this could be biochemical molecules. This could be a lot of different things that you can think of. They go through distortion, textures, dynamical properties, and then get into cholesterics and smectics and what those represent. So this is a lot of book here, but I'm giving this to people as a reference, and it might be a little over people's heads in certain parts, but don't be afraid of that. You just, if you find an area that you like in the table of contents, just go to that area and, read up on it, and, maybe things will start to make sense. It's the way I've typically learned things, and I think that's a pretty effective way to go through those sorts of things. A related paper is this Quantum Magazine article on Turing patterns, and this is Turing patterns turn up in a tiny crystal. So making this connection between Turing patterns, which we've talked about with respect to biology and chemistry, and then a tiny crystal, which is the system that they're going to talk about. So The mechanism behind leopard spots and zebra stripes also appears to explain the pattern growth of a bismuth crystal, extending Alan Turing's 1952 idea to the atomic scale. So we've talked about the mechanism behind Turing patterns, which is this Turing morphogenesis. It's basically where you have all these like molecules, or they could be, what they call morphogens, that are distributed in space. And there are these chemical signals that exist in gradients, and they control how these things are, organized. If they organize pattern, if they organize stripes, if they organize fields of different colors and things like that. And you see this in biological systems. In this case, you're seeing a crystal at the atomic scale, which is much smaller than what we usually think of. And so this is the nanometer-wide stripes seen in crystal growth left, closely match those formed in a theoretical model based on Turing patterns, right? So these Turing patterns, of course, you can implement them in cellular automata. So we can build a computational model. I don't know if that's what they're doing here, but you can build a computational model of this, and you can replicate something you see in nature. And so the idea is that, if we can do that, we can build a model that replicates those mechanisms, and if it works, then we know it's probably working in nature in a similar way. So in this case, several years ago, a team of physicists at Stanford, led by this scientist, was trying to grow a thin layer of bismuth crystal on a metallic surface. But instead of forming a uniform sheet, the crystal became a patchwork of uneven growth. So, they thought that they were going to get this uniform sheet of crystal, but it became a patchwork. In some areas, those where the crystal layer was only one atom thick, a striking design emerged. Small stripes filled irregular patches, and these regions butted against one another. These stripes oriented at different angles. So it's like this image here, where you have these stripes that are sort of irregular, but they're regular in terms of their period, but they're irregular in terms of their shape. So they're kind of curved here, as you can see. And so they couldn't explain the stripes. Then working on a working trip to Paris in 2017, showed them to another theorist who said this is like a zebra. And that's, kind of, if you look at it, you say, well, that's like a zebra. Okay, why is that relevant? And if the stripes were really like a zebra's, then they could be a Turing pattern. So, okay, well, that's a hypothesis. So what happened? Why is this important? As it turns out, zebra stripes have this sort of process behind it called the Turing pattern formation, or Turing morphogenesis. And this is something, of course, Alan Turing, the computer scientist, worked on close to his death. This kind of pattern has since been identified in the arrangement of bacteria stripes on seashells, and even the distribution of human settlements. So, but bismuth sheets seem to be missing some of Turing pattern's essential ingredients. Turing's paper described a theoretical mechanism based on two substances, an activator and an inhibitor that diffused across the different areas at different rates. So what happens is you have these chemical agents that diffuse across this space with all these molecules, and they either activate or inhibit behavior. So it could be like growth, or it could be differentiation, or something like transformation, or something like that. And the idea is that these activators will contribute to a stripe maybe of one color, and the inhibitor will stop that stripe from expanding out. So you'll end up with stripes that are separated by a background, as instead of like having this uneven expression of different growth and non-growth. So you get these patterns simply because these things diffuse at different rates and they interact. The interaction between these two morphogens, as Turing called them, allows one to interrupt the effect of the other, creating a pattern of colored lines on a tropical fish, or something that is rather than a solid color striped. So the activator will override the inhibitor. Sometimes the inhibitor will override the activator, depending on the concentration of each in some location. But in the case of a bismuth crystal, there is no diffusion. Molecules don't shift randomly and spread out while reacting with each other. And then after 3 years, they ended up with a simulated pattern. This is published in Nature Physics. They have a link to the paper here that look almost identical to the stripes in the real crystal. And so this is a nice match of the model and the experiment. And so that's the paper here. What's interesting here, I think, with this liquid crystal perspective, is that we talked about gene expression and gene regulatory networks before, and gene regulatory networks are, of course, one way in which this works. You can have the expression of different chemical agents in a certain location, but you can also just do this with physics. You don't really need to have a genetic system. So this is something, these are two different sort of hypotheses as to how patterns are formed. You can have this sort of regulatory network where things are, that these morphogens are expressed in certain locations. You can also have these self-organizing physical systems where you have, different things at different orientations, and they kind of emerge. So, and of course, a gene regulatory system or something controlled by one of these is also sort of emergent as well, but those are two different mechanisms. So I think that's an interesting thing to follow up on. I know Susan wants to do some things with soft active materials and that. So we should keep that thread going in our meetings. Anyways, I didn't have much more to say about it. I just wanted to highlight some of these things here. Actually, there's another paper in here, and I didn't put it in the folder, but there's this paper, new paper, in biosystems, spatial waves and temporal oscillations in vertebrate limb development. So this is something else. This is where we're talking about spatial waves and temporal oscillations. So this is taking this Turing type reaction diffusion model, or this morphogenesis model, that generates spatial standing waves of cell condensations. So what they're doing here is they're looking at developing vertebrate limb buds as an excitable medium. So an excitable medium is something where the cells have this excitability where they, can transform into different things. And this excitable medium sustains both spatial and temporal periodic phenomena. So this, of course, we're talking about this Turing morphogenesis, this reaction diffusion process that I had mentioned previously, that generates spatial standing waves. These condensations are transformed into the nodules and rods of the cartilaginous and eventually bony endoskeleton. So there's this process where bones are built, or bone material was built out of these cells. The second outcome is that temporal periodicity results from intracellular regulatory dynamics. So this is where you have this gene expression network working to form patterns. And so this is happening at one level, but also this Turing reaction diffusion model is working at another level. So they're basically saying that you have this modulation in one sense, in the intracellular sense, but also outside of the cell, across cells, you have this general Turing type morphogenesis. So here we review the experimental evidence from the chicken embryo, interpreted by a set of mathematical and computational models. The spatial waveforming systems are based on two glycan-binding proteins. So these two galectin molecules in interaction with each other and the cells that produce them. And that the temporal oscillation occurs in the expression of the transcriptional coregulator S1. So you have these different proteins and transcription factors that are playing roles here. So you're putting some biological flesh onto these bones of the model. The multicellular synchronization of this oscillation across the limb bud, so this is across cells, serves to coordinate the biochemical states of the mesenchymal cells globally, thereby refining and sharpening the spatial pattern. So this is, of course, something you see in Turing models where the spatial pattern is sharpened by, the synchronization of different signals. Significantly, the waveforming reaction diffusion-based mechanism itself, unlike most Turing-type systems, does not contain an oscillatory core and may be evolved to this condition as it came to incorporate the cell matrix adhesion that enabled the pattern forming capability. So let me see if there are any pictures in this paper. This is a schematic development of a vertebrate limb. So this is where you start off at day 3 and then move to day 5. As you move, you get more detail here. This is between 3 and 7 days of chicken wing embryogenesis. This is where you're starting to get, so you have these different condensations that are going on in this bud. So this is across cells in the bud. Different structures are exhibiting periodicity in different ways. And then this rose shading is where there's morphogenetic activity. So, you start off with this bud here at the bottom, and then the leading edge here out at the edge is the thing that's expanding and growing, and everything is sort of differentiating outward. So you see that, in day 3, you're starting to get this expansion outward like this, and you can see the zones kind of with different shading. And this blue, I guess, is the mature zone, and then these green and purple areas are things that are expanding and changing. And so you can see that this expands at day 5 outward from the base out to the edge, and then finally at day 7, at the end of this process, you have this limb bud. So you can see that there's a lot. It's a very spatial process as it moves outward. So you can see how that works. So I don't want to get too deeply into that paper. I think that's a good way to, I'll leave it at that. Then we also have this follow-up from last week's discussion on replaying the tape. Yeah? Oh,
before you leave that paper completely,
yeah.
I know that a lung development is necessary for limb development.
Okay.
So you have to have the lung development proceeding properly in order to develop a limb properly. This is from a course I took in dysmorphology. Anyways, it's lung development and kidney development go together, but also limb development. Just an odd fact.
Yeah. Well, I mean, I'm sure that that's there are probably a number of reasons for that, but I think that's an interesting point. I wonder if we can find a paper on that where they kind of link those things together. Because I mean, I don't know if it's like that. There's something going on in the developmental program, or if there's something that's like if they're linked somehow functionally, like in terms of the.
Okay, I've actually tried to find that. The information comes from a Dr. Jane Evans, who was a professor at the University of Manitoba, and I haven't been able to find a paper. I've looked for one. So maybe I need to contact her.
Yeah, that would be great if you could do that. That would be an interesting thing to follow up on. Yeah. So, yeah, that would be really nice. So, yeah, I mean, that's, like, again, that's something we can follow up on as well. So this is following up on our discussion on replaying the tape of life last week and different pathways. This is a paper by David Krakauer, who's at the Santa Fe Institute, and Christopher Kemps, and they're talking about the multiple paths to multiple life. I'm not going to go very deeply into this because it's a pretty involved paper, but so we argue for multiple forms of life realized through multiple different historical pathways. And then we talked about last week about the different, like when you replay the tape of life, if you started over and you allowed life to evolve, once again from its precursors on Earth, would you observe the same outcome? And of course, if you had alien life on another planet, would it follow the same outcome as you see on Earth? You have a different planet, you have a different planetary history. Would it look the same? And of course, a lot of people would say no, but maybe there'd be some commonalities. We don't know. But what they're doing in this paper is they're sort of making this, they're kind of considering this as multiple forms of life. They're just basically saying that depending on the historical pathway, you're going to get different forms of life, and that there are multiple historical pathways one can take. So from this perspective, there have been multiple origins of life on Earth. Life is not a universal homology. In earth science, there's this idea that life, the origin of life, is actually, there have been many sort of starts to life, and we believe that there's like this common origin of life, but that these multiple starts of life were, really early in the history of life, and that there wasn't a lot of branching back then. There was a lot of reticulation of the tree of life where these different starts of life would recombine, and so it's very hard to get that signal. But what happened was the signal that we have now, which is usually like molecular data, DNA, and so forth, point to a universal origin. But the universal origin could be more complex than we would like to think. So there's maybe a lot of potential starts to life. Maybe some of those went extinct, but the starts of life that persisted to the common day were recombining and basically into a universal common ancestor that we can measure today. And so by broadening the class of originations, we significantly expand the data set for searching for life. Through a computational analogy, the origin of life describes both the origin of hardware, physical substrate, and software default function. So we're looking at in the origins of life, we're looking at these different components. We're looking at the physical substrate, which is like, it could be like DNA as a replication standard, or it could be like the phenotype of a single cell organism. It could be, the sort of the environment that it's in. So there's this old idea of RNA clays, I think, that's been discredited at this point, but there's this idea that, life takes life can take many forms of a substrate that where life can replicate and survive. And then software, which is the evolve function. So how do things get inherited over time? So like all information processing systems, so they consider DNA to be this information processing system that self-replicates. Adaptive systems possess a nested hierarchy of levels, a level of function optimization, such as fitness maximization, or it could be some other feature in the life form, a level of constraints, energy requirements, and a level of materials, DNA or RNA, genome, and cells. So you have this sort of imperative for optimization or fitness maximization or something. You also have energetic constraints, meaning if you don't have energy, you can't live or replicate. And then a level of materials, so you need to have some basic material. In a lot of cases, this is DNA or RNA. Sometimes it's both in a cell. That's a compartmentalized thing. And so this is how we're just considering different ways life could have originated. You could start with RNA. There are things called protocells, which they can build in a lab where you have this container that it's filled with RNA molecules, and those are sort of on the boundary of living and non-living. So you can have this sort of system, and this could have been something in early life. And if you're on another planet, say, you might find protocells and maybe protocells that have evolved into other things. The functions essential to life are realized by different substrates with different efficiencies. The functional level allows us to identify multiple origins of life by searching for key principles of optimization in different material form, including the prebiotic origin of protocells, the emergence of culture, economic, and legal institutions. So you can apply this broadly to cultural evolution, to software evolution, software agents, etc. Now, I don't want to get too far down that path, but like in this case, we're talking about, basically the origins of biological life. They talk about the last universal common ancestor. Life should be thought of as a special class of convergent evolution. The multiple origins of life on Earth happen to have a common historical trajectory in Luca. So this is the last universal common ancestor. As has been noted, if new life were created in a computer or in a laboratory, those specific substrates are set up by humans and create a causal link with Luca. So, there's this, and then living across levels, so they're trying to sort of get a handle on these layered or multi-level structures for thinking about life, inspired by Mars levels of information processing for vision, which is something that we talk about a lot in artificial intelligence. And so it's an interesting take on it. It's, thinking about life as a sort of sentient thing that, might be, a lot of people argue about the definition of life. This is another way to look at this. So this is looking at different layers of information processing, but it's a useful analogy for illustrating the type of theory that we want to build in terms of the origin of life. So it's an interesting paper. I think Jesse would be very interested in this paper. And this shows the materials of life, the constraints, and the optimization principles. So you have these constraints, physical constraints we've talked about in the last couple papers. We've talked about engineered constraints, energetic constraints. We have these material possibilities, so you can have life as like crystals, or life as like biological biomolecules, or maybe even rocks or clay. I don't know. I mean, this is the point is that they're saying that you could have a wide range of materials that could possibly be, you could build upon to build a living system, or something that evolves. And so then you have the space of optimization principles, which is the process of evolution, and what they call the space of the living, which is what we observe today. And so this is the history-centric perspective. This is where you have, depending on the history of the taxon that you're looking at, life, the origins of life may be different. They may converge upon a common ancestor. So that's a follow-up to that discussion. And then finally, I wanted to talk about one more paper here, if I can find it. This one here. I think this would be interesting. This is the ethology of morphogenesis, reveals the design principles for Cnidarian size and shape development. So this is talking about morphogenesis again. This is talking about Cnidarians, which are a marine invertebrate, and this is talking about their size and shape development. But I think in this case, they're talking about how behavior regulates that. So the abstract reads, during development, organisms interact with their natural habits while undergoing morphological changes. Yet it remains unclear whether the interplay between developing systems and their environments impacts animal morphogenesis. Here we use the Cnidarian Nematostella vectensis as a developmental model to uncover a mechanistic link between organism size, shape, and behavior. So this is interesting. This is the developmental model where you're connecting behavior, organism size, and organism shape. And of course, we talked about this a little bit with C. elegans, where you have this developing connectome, and then there are behaviors in early development, such as muscle twitching, that occur sort of, autonomously, and then you get connection of this connectome, and then they start to engage in these very basic behaviors, and then the behaviors diversify throughout larval development. And in this case, they're able to look at like the development of the organism itself and the behaviors. So this is a nice model for this type of thing. Using quantitative live imaging, including behavioral profiling, combined with molecular and biophysical experiments, we demonstrate that the muscular hydraulic machinery that controls body movement, which is this idea of muscle being like this hydraulic type mechanism, directly drives larval polyp morphogenesis. Unexpectedly, size and shape development are differentially controlled by antagonistic muscles. So this is where the muscles are controlling the size and shape development in this organism. A simple theoretical model shows how a combination of slow priming and fast pumping pressures generated by muscular hydraulics acts as a global mechanical regulator that coordinates tissue remodeling. Altogether, our findings illuminate how dynamic behavior modes in the environment can be harnessed to drive morphogenetic trajectories, establishing ethology as a critical component of organismal morphogenesis. So this is where they say that behavior, or what they call ethology, which is the study of behavior, can affect morphogenesis. So this is a very different model organism than C. elegans. You have this larva polyp transition, and so this is where they're looking, and they're looking at how you can have these variability in development based on some of these muscular behaviors. And like I said, in C. elegans, the muscles behave in a way that's, I think the pictures are down here, okay, in a way that's autonomous, and then it hooks up to the connectome. In this case, they're largely interested in that sort of muscle autonomy. So you have this. This is the organism here with the oral pole and the aboral pole, which is the two ends of the organism, and they show movement in these different parts, the centroid and then at these poles. So they have two conditions, low motility and high motility. And then this is where you have different expansions. So this is an isotropic expansion. This is an isotropic expansion where it expands along two axes, not just the anterior posterior axis. This OC and then no elongation. And so these behavioral modes sort of correlate with morphodynamics in this way. You have projection of the oral and aboral pole location in a sessile motile animal. Here in this B and C, these graphs are where you have time-shifted distributions of morphodynamics for sessile and motile animals. So this is the sessile animal for these different conditions. This is the motile animal. So a degree of motility and the number of or the percent of animals that do this. So you can see that the anisotropic expansion and the OC and the no elongation are all a greater percentage of animals, and this isotropic expansion is in much lower percentage of animals. And then here you can see the same thing where no elongation dominates in the motile example. So you have this difference between whether they're moving or not moving. The motiles where they're moving, the sessiles where they're not. And so this is an example. This is a picture of the organism in more detail, where they're going from this stage here to the polyp, which is this stage where they have more defined morphology, and this is the elongation that occurs. And so you can see that this is what they're looking at, and so you can see that there are different behaviors based on what the muscles are doing that determine how this expands. Again, this is, so you can see here that you have muscles and muscle fibers. These F-actin molecules are controlling a lot of this expansion of the muscles. So they're doing this, there's this sort of hydraulic mechanism here with the muscles, and it's driving a lot of this expansion. And then they have some molecular experiments where they show some examples of, I think, where they're knocking down some RNA, or they're knocking down some genes using RNA. And so they're actually looking at what the effects of that. And then finally they have this is where you have the size-shape trade-off in this A figure here, where you have the wild type, you have these different mutants, and then an open capillary system where it's arrested. And basically in the wild type, size and shape are regulated. If you knock out this TBX20, shape is preferred over size. This BMP, this growth factor, when it's knocked out, size expands over shape. And so you have these different types of larvae that grow in different ways. And so it's an interesting way to characterize this process. And so that's a behavior driven. So that's all we had to say about those papers. I think that's enough for today. So is that a hydra? It's actually not a hydra. It's a, let me go back to the papers here. It's a Cnidarian, so it's I think it's closely related to a hydra, but the actual species here is Nematostella vectensis, and I'm not familiar with that organism, but it's a it's a Cnidarian. It's pretty close to a hydra, but they look very similar in a lot of ways. And so Shruti had network issues, and so Akshay asked, does it have theories about surface tension of liquid crystals? So I think that, yeah, they do talk about maybe surface tension. They do some experiments with liquid crystals where they, there are certain experiments where where you have a liquid, and then in a liquid phase, the different molecules can migrate out to the edge of the liquid. So they have they do different types of experiments like that. I can't really remember what's in the book, but if you look through the table of contents, you might find some interesting things to follow up on. So that's all for today, I think, unless Susan has any comments or questions.
No, I don't. Not at the moment. I'm interested in the surface tension of the crystals as well. It looks like their 3D modeling of cells that they've been doing for active material.
Yeah.
Like the vertex model. Yeah, they've been working with 3D shapes like that.
That's good. So it's coming along pretty well in terms of the research.
Yeah, I've been looking at it. I'll try to put together a presentation. It's just taking me a while. My eye was out of commission for a bit.
Oh, yeah. Yeah. Well, hopefully you're feeling better.
Yeah, no, it's fine. You notice the lack of glasses. I need new glasses now.
Oh. Okay, well, good luck. Okay, well, thanks for attending, and this will be up later. And if you have any comments, leave them in the Slack or email. And hope to talk to you later this week. If you want to present something for next week, let me know.